At the very tips of every chromosome in every cell in your body, there are tiny protective caps made of repetitive DNA sequences, and every single time that cell divides, those caps get a little bit shorter. Not damaged, not broken, just shorter by design as an unavoidable cost of copying the chromosome. These caps are called telomeres, and if you've never heard of them, they might be the single most important structure in your body that nobody ever told you about because their length is essentially a biological countdown clock, a molecular timer that determines how many more times a cell can divide before it either enters a permanent retirement state called senescence or triggers its own self destruction. Short telomeres don't just mean your cells are old, they mean your cells are running out of time. The analogy that researchers use most often is the plastic tip on the end of a shoelace, the aglet. As long as the aglet is intact, the shoelace holds together and does its job. But once the aglet wears away, the lace starts fraying, the threads unravel, and eventually the whole thing falls apart. Telomeres work the same way. As long as they're sufficiently long, the chromosome stays stable and the cell can divide safely. But as they shorten with each division, the chromosome's essential genetic material becomes increasingly exposed to damage. The cell's ability to function and repair itself declines, and the risk of mutation, malfunction, and disease rises sharply. A landmark study from the University of California, San Francisco, led by Nobel Prize winning molecular biologist Elizabeth Blackburn, who shared the 2009 Nobel Prize in Physiology or Medicine specifically for discovering telomerase, the enzyme that maintains and can rebuild telomeres, found that shorter telomere length is independently associated with a dramatically higher risk of cardiovascular disease, type 2 diabetes, osteoporosis, cognitive decline, certain cancers, and early death from virtually every major cause. When Blackburn and her colleagues measured telomere length in thousands of participants and followed them over years, the pattern was unmistakable. People with shorter telomeres aged faster, got sicker sooner, and died younger than people of the same chronological age with longer telomeres. Your biological age, it turned out, wasn't determined by the calendar, it was determined by your telomeres. But the study also revealed something that transformed the entire field and opened the door to the conversation we're having today. Telomere length is not genetically fixed at birth. It is not a predetermined countdown that you have no control over. It is profoundly and continuously influenced by lifestyle factors, stress, sleep, exercise, and critically, what you eat. Certain nutrients and bioactive compounds found in ordinary food have been shown in published, peer-reviewed research to slow the rate of telomere shortening, protect telomeres from the oxidative damage that erodes them between divisions, and even activate telomerase, the enzyme that can physically rebuild and lengthen telomeres that have already shortened. That last point is worth pausing on. We're not just talking about slowing a decline, we're talking about a biological mechanism for partial reversal, and the foods that support it are available at any grocery store. Food number one is flax seeds, and their telomere protection comes from the same omega-3 fatty acids that make them so powerful for DNA repair. Flax seeds are the richest plant-based source of alpha-linolenic acid, the omega-3 fatty acid that your body partially converts into the longer-chain EPA and DHA, and omega-3s have been directly linked to telomere preservation in one of the most influential studies ever conducted on diet and cellular aging. A study published in the Journal of the American Medical Association followed over 600 adults with stable heart disease and measured their blood levels of omega-3 fatty acids alongside their telomere length over a 5-year period, and the results were striking. Participants with the highest omega-3 levels experienced significantly less telomere shortening over the study period compared to those with the lowest levels. The mechanism appears to involve omega-3's ability to reduce both oxidative stress and chronic inflammation, the two primary forces that accelerate telomere erosion beyond the normal rate of division-related shortening. 2 tablespoons of ground flaxseed daily provides a concentrated dose of plant-based omega-3s alongside the lignans and fiber that we've discussed in our DNA repair video, making it one of the most efficient telomere supporting foods available. Food number 2 is spinach, and the specific nutrient it provides is so critical for telomere maintenance that deficiency alone can accelerate your cellular clock. Spinach is one of the richest dietary sources of folate, vitamin B9, and folate's role in telomere biology is both direct and irreplaceable, making spinach one of the most important telomere supporting foods you can eat on a regular basis. Folate is required for 3 of the most fundamental processes in cellular biology: DNA synthesis, DNA repair, and DNA methylation. And all 3 of these processes are intimately connected to how well your chromosomes maintain their telomeric caps during the thousands of cell divisions that happen throughout your body every day. When the folate levels are adequate, the molecular machinery that copies chromosomes during cell division runs smoothly and accurately, the repair enzymes that fix replication errors work efficiently, and the methylation patterns that protect telomeric and subtelomeric DNA remain stable. But when folate drops below optimal levels, which is disturbingly common in older adults due to reduced dietary intake, impaired absorption, and medications that interfere with folate metabolism, the entire system degrades. A study published in Clinical Nutrition Research found a clear, statistically significant association between adequate folate and B12 levels and longer leukocyte telomere length in the study population, while elevated homocysteine, an amino acid that rises in the blood when folate and B12 are insufficient. And that is itself a direct cause of DNA damage and vascular inflammation, was independently associated with shorter telomeres and markers of accelerated cellular aging. The connection couldn't be more direct. Folate supports the molecular infrastructure that keeps telomeres intact during the single most dangerous moment in a cell's existence, the act of dividing and copying 3 billion base pairs of DNA without error. And without enough of it, telomere erosion accelerates beyond what cell division alone would cause. 3 cups of raw spinach or 1 cup cooked provides roughly 65% of the daily folate requirement, and combining it with other folate-rich foods like asparagus, lentils, and broccoli can ensure your cells never run short of this critical telomere maintenance nutrient. Food number 3 is salmon, and the specific form of omega-3 it provides may be even more potent for telomere protection than the plant-based form in flax seeds. While flax seeds provide ALA, which must be converted before it can be fully utilized, salmon and other fatty fish deliver preformed DHA and EPA, the long-chain omega-3 fatty acids that can be incorporated directly into cell membranes and that have the strongest research support for telomere protection. The JAMA study we mentioned found that it was specifically the blood levels of marine-derived omega-3s, DHA and EPA, that correlated most strongly with slower telomere shortening, and subsequent research has confirmed that these fatty acids reduce the inflammatory signaling molecules and oxidative byproducts that erode telomeric DNA between cell divisions. 2 servings of fatty fish per week provides a clinically meaningful intake of preformed omega-3s that your body can use immediately without the conversion losses that occur with plant-based sources. Food number 4 is blueberries, and their telomere benefit works through the polyphenol pathway we explored in our 30-day blueberry video, but with a specific twist. The anthocyanins in blueberries don't just protect telomeres passively through general antioxidant defense. Emerging research suggests they may actually stimulate telomerase activity, the enzyme discovered by Elizabeth Blackburn that can rebuild shortened telomeres by physically adding new repetitive DNA sequences back onto the chromosome tips. Telomerase is naturally active in stem cells, in certain immune cells, and during embryonic development, but its activity declines sharply with age in most adult tissues, which is one of the primary reasons telomere shortening accelerates after middle age, and the diseases associated with short telomeres, cardiovascular disease, diabetes, cancer, dementia, cluster so heavily in the older population. Polyphenols from berries and other plant sources have been shown in laboratory studies to upregulate telomerase gene expression, essentially reactivating the enzyme at a level that may be sufficient to slow or partially counteract the age-related decline in telomere maintenance, which is as close to reversing the cellular aging clock as anything in nutritional science has yet been able to demonstrate. A daily cup of blueberries provides one of the most concentrated anthocyanin doses available from any common fruit, and the effects compound over time as the gene expression changes, build cumulatively with consistent daily intake. Food number 5 is green tea. And EGCG's effect on telomeres adds yet another layer to the already extraordinary cellular profile of this ancient beverage. EGCG from green tea has been shown to both protect telomeres from oxidative damage through its potent antioxidant capacity and to modulate the epigenetic marks that influence telomere maintenance, specifically DNA methylation patterns at the telomeric and subtelomeric regions of chromosomes. Research published in aging journals has found that regular green tea consumption is associated with longer leukocyte telomere length in population studies, and laboratory work has demonstrated that EGCG can reduce the rate of telomere shortening in cultured human cells under oxidative stress conditions. 3 to 4 cups daily continues to be the intake level most supported by the literature. Food number 6 is walnuts, and they deliver a unique combination of telomere protective compounds that few other single foods can match. Walnuts are rich in alpha-linolenic acid, the plant omega-3, vitamin E, a fat-soluble antioxidant that protects lipid membranes from oxidation, polyphenols, including ellagic acid and the amino acid L-arginine, which supports nitric oxide production, and blood flow. This combination addresses telomere erosion from multiple angles simultaneously. The omega-3s and vitamin E reduce the oxidative stress that directly damages telomeric DNA. The polyphenols support anti-inflammatory pathways that slow the background rate of telomere shortening, and the improved blood flow ensures that the nutrients protecting telomeres in distant tissues actually reach them. A study from Brigham and Women's Hospital found that women who ate walnuts regularly had significantly longer telomere lengths than those who didn't, with each additional serving per week associated with measurably longer telomeres in a dose-dependent relationship. And food number 7 is bell peppers, and the reason comes down to a vitamin most people take for granted. Bell peppers, particularly the red and yellow varieties, are among the richest dietary sources of vitamin C, containing even more per serving than oranges. And a cross-sectional study published in Frontiers in Nutrition, using data from the National Health and Nutrition Examination Survey, found a significant positive association between dietary vitamin C intake and telomere length in adults. The relationship was linear and dose-dependent, and the more vitamin C people consume from food, the longer their telomeres tended to be. And the proposed mechanism involves vitamin C's role as a primary water-soluble antioxidant that protects DNA from oxidative damage during replication, reducing the rate at which telomeres erode beyond what's expected from normal cell division alone. One medium red bell pepper provides over 150% of your daily vitamin C requirement, and because the vitamin is delivered alongside carotenoids and other phytochemicals in the whole food matrix, the absorption and utilization may be superior to isolated vitamin C supplements. Here's what I want you to understand about telomeres and the food you eat, because it's a message that changes how you think about aging itself. Your cellular clock is not set at birth and ticking inevitably towards zero with nothing you can do about it. It is a living, dynamic system that is influenced every single day by the nutrients you provide your cells, the oxidative stress you expose them to, the inflammatory burden your body is carrying, and the lifestyle choices you make from morning to night. Flax seeds and salmon for the omega-3s that directly slow telomere erosion, spinach for the folate infrastructure that maintains telomere integrity during cell division, blueberries for the polyphenols that may reactivate the very enzyme that rebuilds shortened telomeres, green tea for the epigenetic support that protects telomeric DNA from the inside, walnuts for multipathway antioxidant and anti-inflammatory defense, and bell peppers for the vitamin C that shields your most vulnerable DNA from oxidative assault. 7 foods, 7 distinct mechanisms, all working in concert to slow a clock that most people never realize they have any control over. You cannot stop the clock entirely. Every cell division will always cost a small amount of telomere length, and that's built into the fundamental mechanics of DNA replication. But you can dramatically slow the rate at which it ticks. You can protect the telomeres you have from the premature erosion that stress and inflammation cause. And you may even be able to partially rebuild what's been lost through telomerase activation. The most powerful dial you have for controlling that clock isn't in a pharmacy and it isn't in a doctor's office. It's on your plate 3 times a day, waiting for you to use it. Share this with someone who needs it. Subscribe so you don't miss what's coming next. And as always, take care of yourselves. I'll see you in the next one.